All right, now that we got the technical issues figured out, hello everyone. Thank you for coming. My name is Emilie Reas. I'm an assistant professor in the Department of Neurosciences. I'm also the biomarker core co-leader of our Shiley-Marcos ADRC. So if anyone is interested in learning more about the ADRC biomarker resources, we can also discuss that at the end. But today I'm going to be sharing a little bit about neuroimaging techniques and their applications to aging and Alzheimer's disease, while integrating a little bit of my personal career trajectory and some research opportunities I've had along the way as an example. Is it? Okay, so just to give you a little idea of what we're going to be talking about today, I'll be sharing a very high-level overview of various neuroimaging techniques and give some applications to aging and Alzheimer's disease, including some work from my lab. And I'm going to frame this all on kind of the backdrop of my personal career trajectory from an undergrad student to my current role as assistant professor, hopefully to give you kind of an idea of kind of the ups and downs of an example academic career path, and how to navigate all those highs and lows. Okay, so to start with a little bit of background, aging is often accompanied by some degree of cognitive decline, which often accelerates after the age of 60 or 65. However, not all cognitive change with age is normal. It's been estimated that 1/3 of adults over the age of 65 have either dementia or mild cognitive impairment, which can often be a precursor stage to dementia. Now, dementia is caused by various different diseases, the most common of which is Alzheimer's disease, which accounts for up to 80% of dementia cases and currently affects over 7 million Americans. Biologically, Alzheimer's disease is characterized first by the accumulation of these extracellular amyloid beta plaques that appear a couple decades before any symptoms appear. Next, this is followed by the deposition of these intracellular tau neurofibrillary tangles, then by neurodegeneration or brain atrophy, and then ultimately cognitive and clinical decline. Sorry, everyone. All right, so I told you about cognitive aging and dementia. Okay. It's very important that we're able to distinguish normal from pathological aging, but this requires insight into the underlying brain anatomy, function, and pathology. So to measure these aspects of the brain, we can use something called biomarkers. Generally speaking, biomarkers are, as the name implies, metrics of biological health. You guys are probably familiar with common biomarkers such as blood cholesterol, blood pressure, things like that. An MRI scan can be a biomarker. Biomarkers have several very important clinical uses. For example, cross-sectionally, they can be used to detect or diagnose diseases. For prediction, they can be used to assess somebody's risk for a disease or their prognosis if they have a condition. Longitudinally, they can be very useful for tracking a disease course or monitoring a treatment outcome. So neuroimaging falls within this class of and it comprises a set of techniques that measure brain structure, function, or biochemistry. And importantly, neuroimaging is unique in that it provides information about what is happening in the brain. For example, it's shrinking, it's more or less active. It shows where it's happening in the brain. So this is distinct from biomarkers we can get from, say, the plasma or the cerebral spinal fluid. And it also tells us when in life it's happening. So this is in contrast to postmortem markers, which can only be taken at one point after life. Neuroimaging can be conducted multiple times throughout somebody's life. So generally speaking, there's a trade-off in neuroimaging between how much spatial or temporal information you can get. So here I'm showing just some example MRI techniques. This doesn't include all the imaging techniques, but it's some of the common ones that we often use in understanding brain aging and Alzheimer's disease. And generally speaking, if you are able to get, a lot of temporal information about when something's happening in the brain, you have to sacrifice spatial information and vice versa. Okay, so now I'm going to switch gears a little bit and tell you a little bit about my personal history and how I got to this point and how I was introduced to various neuroimaging techniques along the way. And I'm going to, as I take you through my story, I'll introduce you to those neuroimaging techniques in the same sequence that I learned about them. So we're going to start over 20 years ago now. I was an undergrad at UC Berkeley studying cognitive science, and I had no idea what I wanted to do with my life. I was really interested in the mind and the brain. I had no research experience. So I volunteered in a psychology lab where we did some behavioral research on human spatial navigation. I wasn't sold on it. I thought it was a little bit dry. So I thought maybe I want to be a neurologist. So I did some pre-med coursework on the side. I wasn't sure if I wanted to do that either. So I was confused and I decided to take some time off and work in industry. So I ended up working at this company, SAM Technology in San Francisco, where we used EEG or electroencephalography to identify biomarkers of mild cognitive impairment. And I was very lucky. One of my supervisors was this woman, Linda McAvoy. Some of you may know her, but this is how I met Linda McAvoy, and she will pop up at several points throughout my talk. And I bring her up here just as an example of how critical it is to surround yourself with supportive mentors. She ended up being a very influential person in my career path, as you will see. So while I was at SAM Technology, this was my first introduction to neuroimaging. And here I learned about EEG or electroencephalography. EEG has very high temporal information. We can get information down to the millisecond resolution, but not great spatial resolution. It's on the order of centimeters or so. So what is EEG? Basically, it involves placing a whole bunch of electrodes on the scalp. So this is non-invasive. And it measures coherent electrical activity that originates from the brain, specifically from postsynaptic potentials. And here's just an example. Oh, sorry, this is so small. An example of an EEG trace. Each line represents a different electrode, and you can see these rhythmic oscillations that are coming from the brain. EEG outputs various different metrics that can be of interest to us. You can measure power in different frequency bands. You probably heard of delta, theta, alpha waves. And here's an example study measuring the frequency of alpha in younger adults versus older adults. And you can see here that the older adults have a slower peak frequency in their alpha band. So that's actually a well-documented finding. In addition, EEG can be used to measure something called evoked potentials. This is the average electrical response locked to some sort of sensory or cognitive stimulus. And we get these characteristic peaks or troughs of activity that correspond to either sensory or cognitive processes that are linked to that stimulus. Okay, so moving along, sorry, I'm doing kind of a whirlwind through these techniques, but moving along, after my time in industry, I realized that I loved research, that decided this was the path for me. So I was gung-ho and said, I'm going to go to grad school, I'm going to get my PhD, I'm going to be a neuroscientist. So I applied to several grad school programs. I got some interviews, super excited, and then I got rejections across the board, including from UCSD. So, this was really disappointing. But at that time, I knew I wanted to be a scientist. I knew I wanted to be a neuroscientist and said, I'm not going to give up. I'm going to try again. And so the next year I reapplied, and I got a bunch of acceptances. So, the lesson here is that sometimes life is unpredictable. I don't know what was different from my first versus my second applications, but I got lucky the second time, and the second time around, UCSD accepted me. And so I ended up here for my PhD. And I did my doctoral research under the mentorship of Jim Brewer. Many of you are probably familiar with him. He is the chair of the neurosciences department and also directs our ADRC. And during my PhD is when I was introduced to MRI. So my dissertation used functional MRI to understand how a part of the brain called the medial temporal lobe contributes to memory retrieval. And I'm not going to go into my personal research because it's not that relevant, but I will show you a little bit about MRI. So there are actually various forms of MRI. But to start off with, I'm going to highlight structural MRI and functional MRI because these are the techniques I was first introduced to. So broadly speaking, MRI involves a large machine that's placed in a really strong magnetic field. It's roughly 60,000 times the strength of the Earth's magnetic field. And we pair it with some radio frequency pulses. And these pulses polarize the protons in the water in our body. And when the radio frequency pulses turn off, the protons relax back to their normal state. And the rate at which they relax back differs by the type of tissue. So this allows us to identify areas of tissue contrast that creates a picture of our tissue of interest. And so this is just an example picture from an MRI scan that you can get of the brain. So there are a whole bunch of different MRI types. Some examples include structural. This is a structural scan. Functional, that measures an estimate of brain activity. Diffusion MRI measures microstructure. Arterial spin labeling measures blood perfusion. Dynamic contrast enhanced MRI measures blood-brain barrier permeability. And I'll touch on some of these methods a little bit later. But just for some examples of how we can use structural MRI, this paper came out 16 years ago by Dr. McAvoy. I said she was going to come up again, so here she is. And she and her colleagues identified this pattern of brain activity that is characteristic of Alzheimer's disease. Then they took a sample of individuals with MCI and classified them according to whether they had this AD-like atrophy pattern or did not. And interestingly, they found that the group that showed this AD-like atrophy pattern were more likely to decline clinically over the next couple of years. Now, functional MRI is a little bit different. Oftentimes we hear of fMRI as measuring brain activity. This isn't exactly true. So what it's actually measuring is the ratio of oxygenated to deoxygenated blood. And this is useful because when neurons in the brain are active, they're actually coupled to the vasculature. And the ratio of oxygenated blood increases when the neurons become active. So just as an example of using fMRI in Alzheimer's disease, this study identified areas that were functionally connected. So they showed correlations in that bold response across a region of the brain called the default mode network. And they found that individuals with Alzheimer's disease showed lower functional connectivity in this network compared to controls. All right, moving along. So during my PhD, I again faced a personal crisis of what do I want to do with my life. I really loved research, but again, I wasn't sure that I wanted to go down, the tenure track, have my own lab. During my PhD, I got really interested in writing, science writing, actually. I wrote my first papers, I wrote my first grant applications. I really loved it. And at the same time, I started doing freelance science writing for media outlets. And I thought maybe I want to be a science writer. So I was kind of toying with the two ideas. When Linda McAvoy again approached me, she was on my PhD committee and with me as a postdoc. So sure, why not? I don't have a plan for my life. So I put science writing for the side. I was doing it a little bit on the side, but I ended up working with Linda as a postdoc, and I ended up doing a 7-year postdoc, which is a little bit longer than I would recommend. But I really enjoyed my time as a postdoc. It was a fantastic opportunity to get some great mentorship and to really build up my skills. And I was really fortunate during this time to be handed a project that was really cool. So there was this diffusion MRI technique that had been developed here by some of our colleagues, Anders Dale, and Linda and Anders suggested that I use it to understand some of the brain changes that occur in Alzheimer's disease. So in my very first months as a postdoc, I applied for this new investigator award from the ADRC, and I was awarded this pilot grant, which really launched my career, to be honest. Shortly after I collected some data from this pilot grant, I was awarded a grant from the Bright Focus Foundation to do some follow-up work. So this was a really transformative period in my career. And this is when I learned the technique diffusion MRI. So diffusion MRI, it's a little bit complicated, so I'll try not to lose you all here. But it's a technique that allows us to estimate tissue microstructure from different patterns of water diffusion. So for example, if we have water molecules in a big open space where there's just water, the water molecules are going to move very fast and in all directions. If we have water molecules inside of a cell, they're going to be very restricted in their movement, and they're not going to move very fast, but they're still going to move in all directions, just not very far. Similarly, if the water molecules are in a cell, but in kind of long processes like axons or dendrites, they're not going to move very much, but if they do move, they're going to move along that principal direction of the axon or the dendrite. So you can see that water is behaving in different patterns according to where it is in the tissue. So the technique of diffusion MRI that we use is called restriction spectrum imaging, or RSI. And this is a multi-compartment model that measures the direction and the rate of diffusion. And from this model, we can compute various metrics of interest that map onto different tissue properties. So for example, free water is found in the cerebral spinal fluid. Hindered diffusion is found in either the extracellular space or in very large cell bodies. Restricted isotropic diffusion is found inside cell bodies, and neurite density refers to diffusion in those axons and dendrites.
How is this different than diffusion tensor imaging?
Good question. So diffusion tensor imaging is a much more simple model. It can't distinguish, for example, if the so in MRI you have kind of a voxel. It's like a three-dimensional cube. And with diffusion tensor imaging, you only get the primary direction of diffusion in that voxel. Restriction spectrum imaging actually measures diffusion in multiple directions, even within that voxel. So it can tell you if there are, for example, like crossing axons or crossing dendrites inside of a voxel. So it gives us a lot more information about kind of the finer scale microstructure. Okay, so we use this technique, RSI, to understand some of the brain microstructural abnormalities in Alzheimer's disease. And here I'm going to be showing data from a group of cognitively normal individuals and a group with either mild cognitive impairment or mild AD that were all recruited from the ADRC. And we found that The cognitively impaired group in black showed abnormal microstructure, including reduced restricted diffusion or intracellular diffusion and higher free water in various white matter tracts, as well as in the hippocampus and the entorhinal cortex, which are sites of early neurodegeneration and pathology in Alzheimer's disease. Importantly, we also found that many of these metrics correlated with cognitive function and really critically predicted cognitive decline across multiple cognitive domains over a 4-year follow-up period. Additionally, we found that higher levels of amyloid pathology in the cerebral spinal fluid. So this is confusing, but in the cerebral spinal fluid, lower A beta 42 is greater pathology. These correlated with abnormal microstructure, including lower restricted diffusion and higher free water. So we were really excited about these findings. These RSI metrics seemed extremely sensitive to brain changes in Alzheimer's disease that even predicted cognitive decline. And so we wondered, okay, given how sensitive they are, do they also pick up on microstructural changes that occur in normal aging or even preclinical Alzheimer's disease? So my mentor, Linda McAvoy, was collecting RSI on mostly cognitively normal older adults from the Rancho Bernardo study. And so we looked just at simple correlations with age to see if there was an age effect here. And we found these really robust correlations. So here, this is just showing the correlation coefficient for different RSI metrics across the brain for women and men. These correlations were really striking, but interestingly, much stronger for women than for men. Next, we wanted to understand, could these diffusion MRI metrics be picking up on changes that happen in cognitively normal individuals before they go on or at genetic risk for Alzheimer's disease before any cognitive symptoms appear? And we found that individuals who carry one or more copies of the ApoE4 gene, and this is the strongest single genetic risk factor for Alzheimer's disease, these individuals showed lower neurite density in the entorhinal cortex, which is a site of very early tau accumulation and atrophy in Alzheimer's disease. Interestingly, we also found a sex difference here, where those ApoE4 differences in entorhinal microstructure were only present for women. Also among women, we saw a difference between ApoE2 carriers and non-carriers, which was fascinating because ApoE2 carriers are thought to be somewhat protected against Alzheimer's disease. So there's this interesting interaction between ApoE genotype and sex on entorhinal cortex microstructure. Okay, so at this point, I was really, excited about the diffusion MRI work we had been doing. At the same time, I had developed an interest in some modifiable risk factors and particularly interested in vascular risk factors. And so I wondered what's going on in terms of vascular dysfunction in the brain. And a colleague suggested that I look into some MRI techniques for measuring vascular dysfunction, including a technique to measure blood-brain barrier permeability. And I thought this is a really cool idea. And I didn't know anybody at UCSD who was doing this. So I applied for a grant from the NIA for a career development award. And this was a very long 2-year saga of me stressing out over my score that was right at the cusp. It didn't get accepted the first round. I reapplied, and then they kept telling me, we don't know, you're right on the cusp. So after 2 years, I finally got the award, which funded a project to study blood-brain barrier breakdown in preclinical Alzheimer's disease. And so this is where I learned a brand new technique called DCE MRI. We had to build the protocol here from scratch, which was a learning experience for me. But before I go into those data, I'm going to give you a little bit of background about why this is interesting. So sporadic Alzheimer's disease is caused by a combination of both modifiable and genetic risk factors. And among the modifiable factors, vascular conditions such as hypertension, obesity, diabetes, high cholesterol are among the strongest risk factors. And this isn't all that surprising because the brain is so rich in vasculature in order to provide essential oxygen and nutrients to the brain. So one pathway by which vascular or other systemic risk factors could influence the brain is by disrupting something called the blood-brain barrier. And the brain is really unique in that it's the only organ to have this property, which is a multicellular interface between the blood vessels and the surrounding brain tissue. And the blood-brain barrier is really critical because it tightly regulates the influx and the efflux of things like nutrients, amyloid beta or other waste products, immune cells, pathogens, toxins, stuff like that. Now, with aging, there are some subtle structural and functional changes that occur to the blood-brain barrier, and these are much more pronounced in various neurological conditions, including Alzheimer's disease. So we can measure the blood-brain barrier or blood-brain barrier permeability using a technique called dynamic contrast enhanced MRI. And briefly, this involves a series of T1 weighted MRI scans or just structural scans paired with the injection of a contrast agent. And this allows us to measure something called K-trans, which is the transfer coefficient of that contrast agent from the blood into the extravascular brain tissue. And this is our measure of permeability. And so we can plot this K-trans metric across the brain. And here's just an example of an 89-year-old male with mild Alzheimer's disease taken from the ADRC here. And you can see the blue areas are where his blood-brain barrier is healthy. But you can see these red and yellow hotspots where there are some areas of blood-brain barrier leakage. Okay, so over the last 5 years or so, we used this technique to measure. Okay, great. Yeah, so I've been here since I started my PhD. It's a hard place to leave. Okay, I guess I will resume. So we used this DCE MRI technique to understand blood-brain barrier breakdown in the preclinical stages of Alzheimer's disease. And so for the analysis I'm going to show you, we focused on cognitively normal older adults and wanted to look at genetic risk for Alzheimer's disease. So we looked at a cohort of ApoE4 non-carriers and carriers. These groups were well matched on age, sex, and education. The more than half were amyloid positive, which was intentional. We wanted to enrich for preclinical Alzheimer's disease, but they were all cognitively normal, scored very well on the MMSE. And these data have been recently published last year and this year in a paper, including one that just came out by my fantastic postdoc, Serafina Solders. So we found that ApoE4 carriers here in orange showed increased cortical gray matter permeability compared to ApoE4 non-carriers. And when Serafina looked at the regional distribution across the brain, she found that it was really pretty widespread, but followed this interesting distribution of really kind of a superior distribution, especially in the frontal and temporal regions. Interestingly, we thought this could just be different due to the fact that E4 carriers might be more likely to have amyloid. When we adjusted for amyloid status, we found that the effects persisted. So this was independent of amyloid. But we wanted to look at that relationship with amyloid a little bit more anyway. And so we stratified by amyloid negative and amyloid positive. And while we found the same general pattern of increased permeability for E4 carriers, it was, to our surprise, stronger in the amyloid negative group. And this was really interesting to us because this seemed to suggest that individuals who are at genetic risk for Alzheimer's disease show this very early increase in blood-brain barrier permeability before any amyloid is accumulating and before any material cognitive impairment is occurring. So this suggested to us that this blood-brain barrier breakdown might be a very early process along the disease course. Next, we wondered, well, what's the impact of blood-brain barrier permeability specifically on neurodegeneration? Because really that's what matters for cognitive decline. And so we looked at correlations with our RSI measures of brain microstructure and found that higher blood-brain barrier permeability on the x-axis correlated with abnormal microstructure, including increased free water, lower neurite density, lower restricted isotropic diffusion. But we only saw these correlations for ApoE4 carriers, for amyloid positive individuals, and for women. And I'll note here that women actually have twice the prevalence of Alzheimer's disease than men. So these three groups here are all high risk groups for developing AD, which suggested to us that blood-brain barrier permeability it might be linked to early markers of neurodegeneration in groups who are at increased risk for getting Alzheimer's disease. Okay, so moving along here, my research was going great. 2020 hits, and as you all remember, the pandemic hit. So I was right about to apply for faculty positions. This was a big disruption in all of our plans. But it ended up, there ended up kind of being a silver lining to this, as I will get to next. But around this time, we were really excited about our blood-brain barrier findings, and we're brainstorming about ways to extend that project into future directions. And the pandemic provided kind of an opportunity for us to shift our focus a little bit. And so 2021, I was promoted to assistant professor here. And so I realized I needed to get funding to support my lab. I applied for about 10 grants my first year, and I got rejections from almost all of them, except for my first R01. So I was pretty excited about that. All the rejections were disappointing, but I got my first R01. And this R01 was to look at blood-brain barrier breakdown in long COVID and its association with Alzheimer's disease risk. So I'm going to step back a little bit and provide a little context for why we went down this path. So shortly into the pandemic, we started hearing reports of people having something called long COVID, which is essentially persistent symptoms that continue beyond somebody's acute infection with SARS-CoV-2. And among those persistent symptoms included brain fog or impairment in various cognitive domains, including memory and attention. And soon after, there were several studies that came out confirming these subjective reports. For example, this study found that individuals who had had a prior COVID-19 infection were more likely to develop dementia or cognitive impairment a couple years later versus people who'd been infected with a different virus. And so this started raising, an alarm in our minds and other groups that there could be kind of this synergy between the long-term effects of COVID and risk for Alzheimer's disease or other dementias. Kind of supporting that hypothesis, studies came out showing that ApoE4 carriers were at greater risk for severe COVID-19 infections, as well as complications like brain microhemorrhages. And over the long term, they were more likely to experience prolonged fatigue, especially mental fatigue. In addition, I mentioned that women are at increased risk for Alzheimer's disease. We now know that women are actually more likely to experience long COVID, especially women who are either middle-aged or older. So here we have this kind of confluence of overlapping symptoms and risk factors between long COVID and Alzheimer's disease. And so we started to wonder, well, what are the mechanisms of long COVID? Do they kind of overlap or perhaps exacerbate the pathogenesis of Alzheimer's disease? And so we still don't understand the mechanisms of long COVID, but there are various hypotheses that are gaining traction. Among those top hypotheses include vascular dysfunction, viral persistence, including infiltration of viral particles into the brain, as well as inflammation and immune dysregulation. And what's interesting about all three of these kind of top hypotheses is that they could all theoretically influence brain health by disrupting the blood-brain barrier. So this was kind of the premise of our R01, and we specifically wanted to examine blood-brain barrier breakdown and brain microstructure in individuals with brain fog related to long COVID and look at modifying effects of Alzheimer's disease risk. So we're about halfway through this study right now. We just published preliminary data from this project. And so I'll be showing data from 49 cognitively normal individuals and then 27 with what we're calling neurological long COVID. Unfortunately, the groups at this kind of interim stage are not perfectly matched on demographics. The long COVID group is somewhat younger, more likely to be female, and more racially and ethnically diverse. Interestingly, though, this actually matches the demographics of the long COVID population. We did some sensitivity analyses on a better matched cognitively normal group and found that the results were essentially identical, so we don't think it influenced our findings. So to start off with, we found that the long COVID group indeed performed worse on various tests, cognitive tests, including verbal fluency, verbal episodic learning and memory. They were also more likely to meet criteria for MCI. In terms of blood-brain barrier permeability, across all the subjects, we found that the long COVID group showed really widespread blood-brain barrier leakage in the cortical gray matter, white matter, and some subcortical regions. We looked at sex interactions with the hypothesis that women would be more likely to show this long COVID-related blood-brain barrier breakdown based on, the findings that women are more susceptible to long COVID and AD. To our surprise, we found the opposite, that only men showed this long COVID-related blood-brain barrier breakdown, and it was really widespread throughout the whole brain. Next, we wanted to see if there were any differences in brain microstructure. And across the whole group, we didn't find much. We found that the long COVID group showed lower neurite density in the amygdala. But when we looked at sex differences, we found this interesting dichotomy where only women showed microstructural abnormalities with long COVID, most pronounced for the white matter. So women with long COVID showed increased free water in the white matter. Free water is often interpreted as a proxy for inflammation, and this was mostly in the frontal and temporal lobes. Finally, we wanted to understand kind of the associations between blood-brain barrier permeability and cognitive and microstructural properties. And we found that indeed higher blood-brain barrier permeability correlated with worse memory recall, as well as higher white matter free water or inflammation. But we only saw these correlations for individuals who were at higher genetic risk for Alzheimer's disease. And we measure genetic risk using something called this polygenic hazard score. So altogether, we see this interesting sex difference whereby men and women respectively show blood-brain barrier breakdown and microstructural damage with long COVID, implicating different pathological mechanisms, and also this link between blood-brain barrier dysfunction and adverse cognitive and microstructural outcomes only in the presence of genetic risk for Alzheimer's disease, suggesting some sort of synergy there. And I'm almost done. So here we are moving along to the present day. A couple of years ago, I was appointed ADRC biomarker core leader with Doug, who is here. Hi, Doug. And so, as I mentioned before, if you have any questions about the ADRC biomarker resources, I'm happy to field some of those questions. And most recently, we were awarded another R01 from the NIH to extend our blood-brain barrier work, and I will discuss that now in future directions. So this new R01 is applying our BBB imaging techniques to understand the dynamics of blood-brain barrier breakdown with this new class of Alzheimer's disease drugs. They're anti-amyloid immunotherapies, so stay tuned for results from that project. And we're also hoping to extend some of our collaborations with the ADRC and affiliated investigators to compare our MRI markers with some PET measures. And so I didn't have time to go into PET, partly because that's not what I focus on, so I'm not the expert on that, but just briefly, PET or positron emission tomography involves the injection of a radioactive ligand which binds to a biological target of interest, for example, amyloid or tau, and we can capture the amount of tracer that's taken up to create these images. And just as an example, here's an amyloid PET scan for a cognitively normal individual, MCI, and AD, and you can see increasing amyloid tracer uptake with greater disease severity. Here's another example from tau PET showing progressive tau uptake that maps on to these Brock stages, which are determined from postmortem pathology. And so to summarize, hopefully today you've learned that neuroimaging is a very powerful tool that allows us to measure brain structure, function, and pathology in living humans. Different neuroimaging techniques provide complementary information about what is happening, where in the brain, and when over the course of aging and disease. From structural, functional, and diffusion MRI, we've learned that that brain atrophy occurs early in AD, and microstructure and connectivity become abnormal very, very early, even in preclinical stages. Blood-brain barrier permeability has shown blood-brain barrier breakdown linked to genetic risk for Alzheimer's disease and other conditions like long COVID, which could perhaps promote AD pathogenesis. And altogether, neuroimaging is very useful for understanding how both modifiable and non-modifiable factors influence the brain and clinical heterogeneity in aging and dementia. And with that, I would like to thank everyone for your attention, my fantastic lab members, collaborators, funding, and I also appreciate teaching evaluations. Those are also very helpful for our continued career progress. And so we have a few minutes left.